Here are two high-yield USMLE-style questions focused on enzyme-linked immunosorbent assay, its mechanism, and clinical applications. Question 1. A 35-year-old woman presents with fatigue, night sweats, and weight loss. She has a history of high-risk sexual behavior and is concerned about possible HIV infection. The physician orders an ELISA test to screen for HIV. Which of the following best describes the role of ELISA in the diagnosis of HIV? A. Direct detection of HIV RNA in the blood. B. Detection of HIV-specific antibodies in the blood. C. Identification of viral antigens in infected cells. D. Direct detection of HIV proteins on the surface of lymphocytes. E. Detection of HIV DNA in peripheral blood mononuclear cells. Detection of HIV-specific antibodies in the blood. Explanation? ELISA enzyme-linked immunosorbent assay is used to detect antibodies against HIV in the blood in indirect ELISA. The test identifies HIV-specific antibodies, such as anti-HIV IgG, which are produced by the body in response to the HIV infection. It is often used as a screening test for HIV, though a Western blot is usually employed as a confirmatory test. Question 2. A. 25-year-old man presents to the clinic for an annual checkup. The physician performs a direct ELISA test on his blood sample to detect antigens associated with a suspected HIV infection. Which of the following best describes the difference between direct ELISA and indirect ELISA? A. Direct ELISA detects antigens, while indirect ELISA detects antibodies. B. Direct ELISA uses a substrate that directly reacts with the antigen, while indirect ELISA uses a secondary antibody linked to the enzyme. C. Indirect ELISA is more commonly used for infectious diseases, while direct ELISA is used for autoimmune diseases. D. Direct ELISA is used primarily for detecting proteins, while indirect ELISA is used for detecting DNA. E. Indirect ELISA involves enzyme-linked antigens, while direct ELISA uses enzyme-linked antibodies. Answer: A. Direct ELISA detects antigens, while indirect ELISA detects antibodies. Explanation: in direct ELISA, the goal is to detect specific antigens present in the sample. The antigen is directly captured by the antibody-coated plate, and the detection is done by adding an enzyme-linked antibody that binds specifically to the antigen. In indirect ELISA, the test detects antibodies against a specific antigen. The antigen is immobilized on the plate, and the presence of the antibody is detected by using a secondary antibody linked to an enzyme.